very good day, guys. My name is Vasanth. I'm a NEET expert. I train NEET aspirants, and we askIITians kind of help kids to achieve their dream in any competitive examinations, which include JEE, NEET examinations, and other competitive examinations. So now today we are going to see or go through the heredity in evolution chapter, a part of it, which you have already seen in the previous video. Okay, today we are going to discuss about few of the important topics of this chapter, and let's get into the chapter right away. Okay, just allow me a minute, guys. I hope everybody could see the screen. Okay, so today we are going to continue with few topics of heredity and evolution, which involve laws of the Mendel. So when we spoke about Mendelian contribution in the previous video, we were actually given with, I have actually introduced with few laws that he has framed, right? I told you about law of dominance, law of segregation, law of independent assortment. So we're going to see them in detail with few examples and how the DNA is going to express and what is DNA and stuff. And then after that, we're going to see a little about blood grouping, how that is going to be different and how complex it is and how what causes the complexity. And then after that, the sexual determination, how the determination of the sex of an individual actually takes place. So these are the topics that we're going to cover in detail in this video. Okay, let's get into the first topic. The first topic states the laws of Mendel. So what is laws of Mendel? It is not the law that which Mendel lived upon. The laws on the basis of which, okay, the genetic expression or the genetic genes function, okay, which has been framed by the observations that Gregor Johann Mendel observed on the basis of his experiment, which we discussed in detail in the previous video. Okay, so it actually contains three laws. Okay, the laws of dominance, laws of independent assortment, and laws of segregation. Previously there were four, and now they are narrowed down to only three of it. So the first law of inheritance is the law of dominance under hybrid condition. The hybrid condition is where an example could be capital T small t, which includes a heterozygous condition, a dominant allele and a recessive allele together. In this case, the phenotype, the expressing character would be the dominant character. So in the hybrid, okay, in the hybrid condition of an offspring, only the dominant trait will be expressing their characteristics. In this case, the phenotype of the plant is capital T small t, that is heterozygous tall, and the genotype of it is heterozygous tall, and the phenotype of it would be tall. So that's the external thing that we would see. So the organism would be taller. Okay, so this is law of dominance. Under heterozygous condition, only one allele which gets to be a dominant. The suppressed allele, the allele which gets suppressed because of the dominant allele, we call it recessive, recessive. So this is how he had determined the character whichever dominated dominated and expressed even in the presence of another allele's existence. Okay, so that is called dominance. So law of dominance states that even at hybrid condition, the characteristics that are able to express, okay, their characteristics even at heterozygous condition is called is what is given away in the law of dominance. That's the first law of inheritance. And the second law of inheritance is law of independent assortment. So independent assortment is like that. When a pair of trait segregates independently, so which means again we'll take the heterozygous part, capital T small t. Okay, in this case, the gamete during the process of gamete formation, okay, the traits will be differentiated and they'll be forming two gametes. If it is going to be capital T, they will again be forming capital T gamete and capital T gamete. If it is going to be small t or dwarf homozygous, then it will be small t one gamete and small t another gamete. But in the case of heterozygous, it will be capital T one gamete and small t another gamete. So these are the this is what is represented in the independent assortment. And a pair of traits segregates independently of another pair during gamete formation, giving different traits equal opportunity to occur together. So this is actually independent assortment. Independent assortment is usually seen in the like, you know, dihybrid cross, because this could be an example, but which could be like, you know, easily understood is capital R, capital R, capital Y, and small r, small y, small y. This is the law which actually states that the allele, the gamete which will be formed will be capital R, capital Y, and small r, small y. So when they are again segregated, it is obvious that the characteristics will be reformed according to the combination or the concentration of it available. The third one suggests that law of segregation. The third law suggests that during the process of gamete production, two copies of each hereditary factors segregate to the offspring, acquiring one factor from each parent, which means, so the gamete which will be formed from a heterozygous plant will be capital T and small t, okay, and another parent could be homozygous. So let's assume this is the male parent and this here is the female, okay, parent. So capital T will be fusing with one gamete of the capital T from the female, so that forming homozygous stop. Capital T will be again. So if you see the gamete from both the parents, one from each is being accumulated. So this results in the segregation, no matter how much, like, you know, they are united, so they are always segregated. So they get segregated during the process of gamete formation. This is what the law of segregation actually states. So these are the three important laws that Mendel have actually like, you know, conveyed, and this, on the basis of these, like the complete genetics has been understood. So there are certain cases where the Mendelian laws were not obeyed. We will be studying about those details in the upcoming classes, especially in 12th standard. Okay, not right now. So next thing that we are going to talk about is DNA. DNA is also called as deoxyribonucleic acid. That's the acid. So that's a expansion of DNA, and it is a molecule which contains genetic codes. So genetic codes are available in the forms of, in the sets of 3. Okay, the sets of 3. So these genetic, with the help of these genetic codes, it literally codes for a character. Okay, that literally codes for amino acid, and then this amino acid codes for another amino acid, and then another code will be available that will be coding for another set of amino acid. Okay. So this is how the complexity is like, you know, widen, and then the characteristics are being expressed. So here there will be another amino acid formed. So if you see the message from which the DNA is supposed to, when which the amino acid or protein supposed to be formed is present in a sequential form in the DNA. So DNA acts as a medium of transferring knowledge from one type of material to another type of material. So it just converts the message that is stored in the form of DNA in the sequences of nucleic acid and get converted into amino acid sequences. So this amino acid sequences will later get formed into a complex protein, and that protein will modify its structure and functions mainly as enzymes. So most of the time, for like, you know, the proteins formed will be functioning as enzymes and other structural cases. Okay, this is the function of DNA. DNA is the most complex material. It is fragile in nature, it is acidic in nature, and it is double helical in nature. So what is double helical? Helical is the swirl that it actually takes place. So it will be double stranded. So the helical structure will be formed like this. So it is usually like this. Okay, that's the double helical structure. So with this, we are okay to go with DNA. So DNA has much complex mechanisms, and we are going to see them in the upcoming slides. So DNA after that. So how the DNA is going to express? So that is also known as central dogma. We call that CENTRAL, central DOGMA. Central dogma is a principle in which the DNA will get transcripted to RNA. Because why is it called transcription? I'll tell you. DNA contains purines and pyrimidines, right? Purines are adenine and guanine. This is the purines. And pyrimidines involve, okay, thymine, and cytosine, and uracil. So these are the three. So there are three pyrimidines. Yes. So in the case of DNA, okay, in the case of DNA, it is adenine, guanine, okay, and thymine and cytosine. So this is the 4 nucleotide bases that is available. But for RNA, okay, it is adenine, guanine, uracil, and cytosine. So in this case, the thymine is replaced with uracil. So that's the modification that actually do. So from the DNA script to RNA script. So transfer of knowledge from DNA script to RNA script is called transcription. Okay. And now the next part of RNA to protein. So RNA, I have already told you the nucleotides are triplet. Okay, 3 nucleotide sequence. Right? Sequence. So we call it triplet codons. Triplet codons. So this triplet codons will be coding for an amino acid. So now here from the nucleotide, from the genetic script to the amino acid, okay, a language. So that's likely that's like completely different. Here there is just scription, like complete, it is transfer, like modification. But in the case of protein formation, okay, in the case of protein formation, it will be complete modification of nucleotide language to protein amino acid language. So that's why it is called translation. The complete language gets transferred so that it becomes a functional protein and then makes the possible things. So what is this red color thing that is actually having an a star over here, and then we mentioned reverse transcriptase? Reverse transcriptase or transcription is a process where RNA, okay, from the RNA, DNA could be formed. So that is another process which you will be studying in higher classes. And replication is where from the existing DNA, new DNA is formed. So this is how the DNA is expressed and how it is sustained in the process of living. Okay, and whenever the cells multiply, it is actually the replication process through which the genetic material is duplicated. Okay, number of copies could be created. So this is the gene expression in nutshell. Okay, so central dogma is very important, in which DNA gets transcribed to RNA and RNA gets transcribed to protein, and the process of conversion of DNA to RNA is transcription, and the process of conversion of RNA to protein is called translation. And the protein undergoes certain modifications to become functional and performance function. So this is the this is how the gene expression actually takes place. Okay, so let's go to the next topic. So next topic literally will help us understand like how the genetic probability actually takes place. So basically there are 4 blood groups. Okay, that's what people always say. Basically there are 4 blood groups. So they are all based on, okay, 3 important things. So one is A allele, okay, B allele, and O allele. Okay, these are the 3 different types. 3 different types. Now, there are 2 alleles that represents the character, right? So here there are like 3 alleles. So on these combinations, it actually forms different types. On these combinations, they form different types. So here, if we apply Punnett square, this will be easy for it to understand. So which I have already done over here. So which we can see very easily, or we can do it also. So if I apply IA, IB, and I. Okay, here it is IA, it is A, B, A, I, and here it is A, B, 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 O, A, I, B, O, and O. Okay, so if you see, This is homozygous. Okay. So how many A's do we have? We have two types of A. One is homozygous A and heterozygous A. Okay, because A and B, both of them are dominant in nature. Okay, and here there are two types of B. B homozygous and heterozygous. And again, how many types of O are present? O, there is only one, because they are both recessive in nature. They could express only when they are in homozygous condition. So that's why they are in nature. Okay. So here we have how many? You can see one homozygous. Okay. 1, 2. Okay. 2. AB, and then we have one homozygous B. Okay. And we have one heterozygous A. And two heterozygous A and two heterozygous B, and then it will be like this. So in nutshell, there will be one of homozygous A and one of homozygous B. There will be two of heterozygous A and two of heterozygous B, and there will be one of homozygous I. Okay, so which totals to 3 plus 3, 6, 7. Okay, totally there are 7. So what about the one that that is actually missing over here? We have not got into one important thing. So what is 9 minus 7? 9 minus 7 is 2. So what was the two things that we are missing? It is because of the coexistence of A and B. So there is a possibility of this too. So there are two of this. So these are the possibilities of groups. So now you think that's it. So you think like, blood groups could be A, B, O, AB. Definitely not. There is another factor, there is another characteristics that is present in the body. Okay. We call them rhesus monkey factor, RH. Rhesus monkey factor, RH. So this factor is found in the rhesus monkey and it is also found in human being. And presence of this factor makes it different. So if the person has A blood group, okay, there are two possibilities. One is A positive and A negative. And if the person is B, there could be B positive and B negative. And if the person is AB, okay, there could be AB positive and AB negative. And if the person is O, okay, there could be O positive and O negative. So if you see, there are basically 4 there were, and now there are, because of the presence of the rhesus monkey factor, now they have doubled. Okay, there are A, B, AB and O. So which means there are 8 blood groups that is possibly formed. So this is phenotypic. So when you know genotypic, there is homozygous A, heterozygous A, homozygous B, heterozygous B, but there will be AB together, okay, and then O, okay, which is always homozygous. So these are the blood groups that is available in the human being. And with this blood group, you need to be very careful because the protein which is present in the blood group. So when you say A blood group will have A antigen and B antibody, okay, and B blood group will have B antigen and A antibody, and O blood group will have, okay, will not have any antigen, no antigen, and both A and B antibody. In the case of AB, it will have both A and B antigen, but no antibody. So what is this thing? This is actually to protect any infiltration. So when a person who has A blood group, when they are diffused or infused with B blood group, so the antigen A will be combining with the antibody A, and that will cause a catastrophic event like blood clotting takes place. So this is one reason why the human being, or like, in human being, no, not only human being, a lot of organisms have blood drooping ability. So this blood drooping had to be taken care. If not, the person will definitely have their blood clot completely. We call that coagulation. COAGULATION, coagulation. So this is the important part. Okay, so blood drooping part. So I hope every one of you are understood about the blood drooping part. So let's go to the next topic. So next topic is sex determination. So determination of the sex, okay, modification of the sex is done by one who has the contribution that could be altered. Okay, whose contribution could be altered. So usually there are 46 chromosomes, right? Chromosomes. So in those 46 chromosomes, 22 pairs, okay, are autosomes, and that one pair is 6 chromosomes or allosomes. Okay, so this makes 23 pairs or 46 chromosomes. So in this, during the sexual differentiation, a male will have 44 plus X and Y. This is for a male. Okay, male. So, but for a female, it will be 44 plus XX. So if you see, when the gametes are formed, it is always in female, okay, it is always 22 plus X. But in the case of male, there are two possibilities. 22 plus X is also possible and 22 plus Y is also possible. So in this case, when the X fuses with X, it is a girl child. Okay. When the X fuses with a Y, it is a boy child. So which organism determines the sex of the individual? It is actually the male who has both X and Y chromosome. So depending upon which one of the sex chromosome possessing gamete fuses with the egg, the sex of the individual is determined. So it is in human being, it is the male which who actually gets to determine the sex determine the sex of an individual. That is not done mandatorily by the by human being, or sorry, it is not done manually by the human being. We cannot alter it. It happens naturally. It is totally random in nature. So this is all the characteristic features of this is what you should know about sexual determination. And all these chromosomes are which is going to be very helpful in determination of the sex of the individual, and all the multiple characteristics that is going to be expressing, which are all sex linked. For example, certain characteristics are sex linked. If you have 2 X chromosome, and certain characteristics will be expressed in extra super, and then when you have X and Y, certain other characteristics for male will be expressed. So this probability is always important. So this is what you should learn about sex determination. So with this, we'll go into some of the few questions which people usually ask. So what is DNA made up of? DNA is made up of nucleic acids. So nucleic acid, we call it nucleotides. So we have already learned the DNA consists of purine and pyrimidine. So adenine and guanine is actually the purine, and thymine and cytosine, okay, and uracil. Okay, uracil. All 3 of them are pyrimidines. In this, let's rule out uracil, which is actually found only in RNA. So the purine always bonds with pyrimidine. So A always bonds with T with a double bond, and then G always bonds with C, or C always bonds with G with a triple bond. So this is what constitutes the DNA. So this is not the only thing that constitutes the DNA. The nucleotide involves, this is nucleotide, this is actually the nitrogenous basis. Nucleotide also includes a deoxyribose sugar plus a phosphate backbone, phosphate molecule. So this literally contributes and forms the structure of the DNA. So this is what you should understand when it comes to the structure of DNA. Next one is, what is the rarest blood group? So rarest blood group is very easy. Okay, it is always the AB negative blood group. So that is actually very rare to form. So we have only 1% of our donors, like, who actually have it, but this is like old data. Now we have more than that because of the huge population increase. But the probability of having AB negative is very, very less compared to other blood grouping. So that's why people who always have AB negative blood group, they are suggested to have, they have A and B. So they cannot be given with any of it. So that's why they should be very, very careful when they undergo blood transfusion. And it is advised for them to, donate blood whenever they could possibly donate. And then you can get the same blood group from the place where you donate when there is a requirement for you. Anything might happen. You should be very careful when it comes to this something like this. Okay. So this is what you should remember. So this is about the rarest blood group. So with this, we will conclude. And thank you so much for your attention. I hope you have enjoyed the class. And if you have any questions, we have a separate portal where you can actually post all your questions, and you will get definitely answers in a jiffy. And expert will be answering, not just any person. And we have exclusive team for that case. And don't stop yourself with just the recorded videos that we are seeing now, and come and join us online, so you would get and watch interactive classes, and you will be getting everything in a fun manner. Okay, thank you so much for your time, guys. Have a nice evening.